All right. I wanted to remind you guys on Monday, but I forgot. So I'm going to remind you now that the discussion board was due for Monday, right, for introductions. We have 27 people enrolled and it only says I have 26 to grade. So that means maybe one person forgot. So try and get that done today because I'm going to start grading tomorrow. And I'm not going to regrade once I start. So remember, it's here under discussions, introductions. Yep. And for it, remember, you have to do your little kingdomology personality quiz, post your results, tell me if the results fit you or not, share any information you want, at least tell me what major you are, or tell us, I should say. And then you had to respond to 2 classmates. So if you're not sure if you did that or not, go ahead and click on your grades. I can't click there right now. I'll show everybody's. Let me go to my student view. So different ways that you can get there is click on grades and click on the introduction, and it will show what you posted. So of course there's nothing there because it's the test student, my practice student. But for you, you should have your initial post that shows up there and 2 replies. If you don't, make sure that you fix that today. Yeah, sure, you can respond to me. I would like for you to interact with your classmates though. But it's okay if you responded to mine. I'll count it. Any other questions regarding that? Okay. So one other shop type. Trying to think how I want to say it. We had our departmental meeting on Monday. We have departmental meetings every Monday. If you look at my schedule, they're even on my schedule. They're getting a little insane about our time. Oh, it's under my welcome. I usually put it under my syllabus and stuff. Does it make sense to be under welcome, or should I just repost it? I'll repost it. Having it more than one place is good, right? But I'm going to change it and post a new schedule for my office hours today. And that's why I was chasing Dr. Coleman down the hallway, because I wanted to find out when our meetings are so that I don't put office hours and then have to change my schedule again. So I had it spread out, my office hours, like basically almost every day except for Tuesday. I'm going to stack my office hours on probably the first 3 days, Monday through Wednesday. There's a reason for this. That does not mean on Thursdays and Fridays where I don't have any office hours that I won't meet with you. I have classes. I'll be here, right? If you need to see me, contact me. We can meet up, right? You all have my Google number. You can text me. You can call me. During the day, if I'm not teaching, I will answer it. Okay? Does that make sense to you guys? All right. But I'll have my little notebook writing down that I met with you outside of office hours too. So don't get freaked out by that. It's just cover your butt type stuff. Make sense? Questions, problems, concerns before we get started on lecture? All right, doing a couple deep breaths. Y'all look asleep.
One quick question. I noticed there's flashcards in the web section. Like 900 of them.
Oh my God, yeah, I guess.
Is that for the whole course or just for the...
I have no idea. I have not been fully trained in Wiley Plus. So I don't, you guys are exploring stuff I haven't even looked at yet. So I don't know the answer to that.
You have no assignments have been issued
Yeah,
yet?
no, that's the other thing I'm going to be working on Thursday. That's the first thing I'm going to be working on Thursday. So expect by Thursday, Friday, you're going to have your first assignment in Wiley Plus. So thank you for asking that because I forgot. Anything else? And there'll be links directly in Canvas to those assignments. So make it nice and easy for you.
So, like I'm looking to get to the part where it would ask me questions.
Right, yeah.
Do you, but it's still not doing that.
No, It's not going to yet. I have to set that up. And then a lot of the stuff we've been covering so far, notice it's not on all of it's in your book, or it's in different sections of your book. So I'm having to like write my own questions and add them to Wiley Plus, and that takes time. And find them in other sections and other places. I'm almost done. Yes, yeah, I agree. I want it to be right before I give it to you guys, and then you're all complaining about it not working properly. But you've had plenty of reading, right, to keep you busy. That you can definitely do through Wiley Plus. All right, so I opened the next one in hopes that I might get to it.
Wiley Plus like practices in it, right?
Yeah, that's what he's asking, like assignments, homework assignments that I'm going to give you guys in there. Like quizzes that you can take. And this is important. I set them up as homework because you're going to do them outside of class, right? But you should treat them as if they're quizzes. Close your notes, close your book, see how much you actually know from what you've been studying, right? Because at this point you should have already started studying, right? You should have a pretty good understanding of those diseases we talked about, right? So that when I post these, you could take it as a quiz. And this will give you an opportunity to go, oh, crap. I studied that and I thought I knew it, but clearly I didn't because I just got all those questions wrong. But what's the good thing about my homework? You can retake it. Do you retake it right away? No, because you didn't know it. You need to what? Figure out how to study it better and then retake it later. Yeah. Yeah. Oh yeah, I think in Wiley too, like in some of the other ones, like if you miss a question, it'll like kind of direct you to where you would find that information in the book. But again, for the questions that I'm going to be adding, you're going to have to look at your notes. Does that make sense?
And is that for a grade?
Yes.
The assignments are.
Yes.
The practice quizzes are not.
Well.
Right now there's read, study, practice.
Oh, read, study, practice is just a built-in thing. You don't get any points for that. Like what he was saying, I think that's where the flashcards are. So, I mean, if you find stuff in there that help you, that you're welcome to use that. We don't have the adaptive program for this book that some of you guys have had. If you had A&P, we don't have Orion. And you guys have taken anatomy and physiology and done Orion. Like, we don't have that. Yeah. Hopefully the next edition of this book it will. I heard it's awesome. So, something I'm looking forward to. Okay? So, yeah, those assignments are graded, but you can do them, multiple times to get a perfect grade. If you look at your syllabus, it's 140 points worth of Wiley Plus assignments. I usually do 14 assignments, 10 points apiece. So we have been on our topic of microbiology. We went over the domains, we introduced viruses, and we were talking about Anthony. We were just about ready to start spontaneous generation, right? So they really, in your book, only talk about Louis Pasteur's experiment with the swan neck flask to help disprove spontaneous generation. I want to talk about a few other people that led up before him that they don't discuss in your book. So again, in general detail, matching type question on your homework, which will be coming up, and on the exam. Oh.
Matching.
Yeah, matching.
Dates. What about it?
No, it's not a history class.
Can you say again what you said about matching?
Yeah. Okay, let me go to the next slide. So all of these guys we're going to talk about In your book, the only one that's talked about is Louis Pasteur's experiment. So you need to have a general understanding from the lecture that I give today about each one of these guys and be able to match them to a description of what they did. For both the homework and the test. Louis Pasteur. So again, you'll notice there's dates here, right? So he asked, do we have to know the dates? No. Do you guys remember last time why I said there was dates next to Anthony's name? To give you a timeline, right? To give you a timeline of how long ago this was. So you'll see they're in order. So we're going to talk about 5 different things. So remember last time we kind of defined spontaneous generation. Anyone want to share what they remember on spontaneous generation? It means it came from where? Out of nowhere, right? So somehow you're ending up with living organisms from non-living things, right? Or decomposing matter, something that was once living. We know that that's not true, right? Life comes from life. It doesn't just spontaneous generate. It doesn't just poof. Magicians can make it look like that, though, but they're just tricking you. So, Francesco Redi did a pretty simple experiment where he took meat and put it in jars. One set of jars he left exposed to the air and the elements. So flies came by. They laid their eggs. Their eggs developed into maggots, the larval form of a fly. Before that, and before his experiment, most people believed that the meat actually turned into maggots. We now know that the flies actually lay the eggs, and those eggs are kind of really small. So unless you're really looking really closely, you would probably miss them. So to demonstrate this, he took another set of jars and he sealed them. Did he end up with any maggots? No, because the flies couldn't get to the meat. Why are the flies laying their eggs on the meat? Yeah, to feed the babies. So when the babies hatch, the food's right there. Because she doesn't stick around, right? She just lays the eggs and off she goes. The next set, he put gauze. You know, like cheesecloth. So there's holes in it, right? So the mom can smell, right, or sense that meat in the container. So she lays her eggs on the gauze. So they're in close proximity of a food source. And of course, the maggots develop on the top of the gauze. Again, separate from the meat, right? That you could clearly see that it is not the meat that's turning into these maggots. Most people accepted this. They're like, oh, okay, I see this now. But that's for something that's relatively large. What about microorganisms? What about that apple juice container I talked about just as we were leaving on Monday that got shoved to the back of the refrigerator and all of a sudden became really cloudy? We now know that that's microorganisms that have grown to large numbers. So much so that it has made that liquid cloudy. As it relates for microorganisms, again, before people knew about them or had an understanding about them, they thought that that apple juice, you know, just turned into stuff. Not that, how do you think those microbes got into that apple juice? Now, the apple juice is food, just like it's food for us, right? For them. Where did those microbes come from? When you open the container. Yeah, from the air, right? So you open, the minute you open that container, what does it tell you to do? Refrigerate it. Because you just let microbes in, no matter what. They're everywhere, right? Why do we put them in the fridge? To slow them down. Yeah, slow them down.
Is there like a phobia against microbes?
Yeah, some people, because we talk about all the things that they could potentially do to us, right? The bad stuff. But didn't I not start this course out with telling you about how awesome they are and how we wouldn't exist without them? We tend to forget that though, right? Just like the news on TV. I think they should be required to have a good segment before the bad. Or just forget about the bad altogether. But that's not what people focus on, right? They focus in on the bad stuff. Right? So for, large organisms, right, that was pretty easy for the flies and the eggs. Could spontaneous generation be true for microorganisms? We know this is not, right? We know where those came from. And even worse, right, if you have someone in your household who decides to drink directly from the apple juice container, their spit, right, their saliva, there's no way microorganisms from them is not getting into that container. You do that in my house, your name gets written on the jug, and it's now yours. You can keep your own microbes to yourself. There's no way that that's not gross. And for some of us, right, like I said, it's very obvious, right, that that's microorganisms. We're not going to drink it because it potentially could be harmful ones. It could potentially make us sick. If there's evidence of a very large number of microorganisms, we're not going to mess with it.
I don't know if you want us to remember Redi by meeting containers. Is that it?
Yeah, well, what would you remember for Redi? What do you think would be important? That he disproved spontaneous generation for larger organisms like flies, and in fact help us know that what? Where'd the maggots come from? The flies whose eggs hatched, right? So he helped make that link that the flies laid the eggs, which led to the maggots, not that the meat turned into maggots. So thus disproving spontaneous generation for larger organisms. Not for microorganisms, right? So again, imagine a liquid food, right, like that apple juice, full of microorganisms. If we boil it, what would happen to those microorganisms? It would kill them. They would die. Do we know if they would all die? No, right? Boiling does not lead to what we call sterilization, which is devoid of all microorganisms. But it will kill most that will harm you. Notice I said most. So why is it every time our water is potentially contaminated, they tell us just to boil it? Because again, most of the ones, right? Yeah, most of the ones that are going to harm you that would potentially be present will be killed by boiling. So you're not sterilizing your water, you're disinfecting your water when you boil it. Hopefully stopping infection. You don't necessarily need your water to be sterile. Still with me? So, John Needham took some liquid food. He boiled it to kill anything potentially that was in there, right? Because under their understanding then, you could kill it all with boiling. And then he let it to cool, but exposed to the air. So guess what happened? Over time, microorganisms from where? From the air fell into it, and they grew in that food source. So did he help disprove spontaneous generation? No, because he didn't stop the growth. He didn't identify where the growth was coming from, right? If anything, he made it a little bit harder. Because he's like, look, they just poof. So Lazzaro came along. He boiled it, but then he sealed it. So no growth happened when it was sealed. When he opened it, though, growth happened. So again, we know nowadays the reason why you got growth after you opened it is because why is being opened bad? Yeah, microorganisms from the air fell into the food source and grew. So why didn't it grow when it was closed? Couldn't get in. No air, right? Well, some scientists would argue, well, of course it didn't grow. There was no air. Because do some microorganisms need components of air in order to grow? We do. You stick us in a sealed container with no air, what's going to happen? We're going to die. What do we need that's in air? Oxygen. Do some microorganisms need oxygen to grow? Absolutely. Some of them are aerobic, just like us. Do all microorganisms need oxygen to grow? There are some that are called anaerobic, and in fact, oxygen would kill them. But of course, most scientists, again, are like, well, the reason it didn't grow is because there was no air. Right? So, Louis started trying to solve this whole air problem. So he filtered air through some cotton and put it into sterile broth that had no growth in it beforehand, food for the organisms. He put that cotton in and he got growth. So he said with that, look, there's some vital force in this air that I trapped in this cotton. And that's where the growth is coming from. Right? There are these particles in the air that we can't see that are living. And when given a large food supply, like what we do in lab, you can grow them to large numbers like that apple juice in the back of your refrigerator and actually see them macroscopically without a microscope. He didn't just stop there, though, right? He made some fancy swan neck flasks, right? And they're not quite as elegant as this picture. But they had at least two curves to them. So this solved the problem of air getting in for oxygen for organisms that may need it. But it stopped what from getting all the way to the food? It stopped from getting in. Notice he has no growth. As long as he kept that sealed and the air had to enter in indirectly, he got no growth. The organisms. The organisms can't fly through the air. There's no flying bacteria. Like, they can't direct what direction. They go on the air currents, right? They're stuck to the dust in the air. They're really fine particles you can't even see. But again, they can't motor on their own. They can't fly. So when they come in, they get stuck in that first bend right there. Right? They get stuck right here. Where the air continues on, right? But unless you're blowing a fan and blowing the dust up this tube, just regular circulating air, that dust is going to settle out in that first bend that the microbes are attached to, and it's not going to get to the food source. So you can leave this stuff out forever, and no growth will happen. And then, of course, he proved that, well, you know, the food I had in there was good because I can break this, open it up to the air, and the microbes in that air can directly fall into the food, and he got growth. Now, again, how did he make his food sterile? Or did he? He boiled it. So really, was there no microorganisms in there? Not any that aren't killed by boiling, because then he would have got growth, right? Does that make sense to you guys? So anybody know a type of bacteria or a form that some bacteria can form that you can't kill it by boiling it? One of the diseases that we talked about on our list does this. Yes, anthrax. Remember the fine powder, right? That wasn't the living cells, it was what? They're in a dormant state. What do we call that? Some people say spores. The true term is endospores, right? Sometimes I'm a bad scientist too and I just say spores. Then that's confused with other spores, right? And it also sounds like reproduction, doesn't it? But as we'll talk about, endospores are just a dormancy period for that cell. The bad news is we can't kill them in that dormancy phase when they're endospores by boiling them. High pressure steam in an autoclave is the most effective way of destroying them. Basically a pressure cooker. Because that spore coat is stabilizing and protecting the DNA. Unless you destroy that DNA, it can become that cell again and actively metabolize. And the bad news with anthrax is it eats you alive, basically.
Because it's so hard to kill. That's why.
Well, no. The thing is, once it gets into your body, it's your immune system can't fight it. It grows faster and is more aggressive than you can fight it. And it eventually will win if you don't get treatment early enough. The people, when they mailed that out in the mail, that terrorist, whoever it really was, some of the people that were exposed, even though they were treated, they weren't treated fast enough and they still died. It's considered a bioterrorist agent because of that. What is anthrax? Bacillus anthracis. Doesn't have virus in the name, does it? So it's probably what? Bacteria. And again, bacteria are the only ones that form these endospores. So he even did stuff where he didn't even break the flask, but even just tipped it, right, and exposed that dust to the food, and he got growth. So he even proved that, he trapped in that bend of the flask the microorganisms that we weren't seeing with our own eyes. So did everybody believe him and they're all like, spontaneous generation is false, there's no way it can just come out of nowhere, everyone agreed, everyone was happy, we moved on? Yeah, no. Humans are just not like that. We don't seem to want to believe anything unless we see it ourselves, right? So what did other scientists try to do? Disprove it. They tried to repeat his work too, right? And they didn't get the same results. So what's going on here? Why didn't they get the same results? Could have been something else in the flask, right? Because what did we say doesn't get killed by boiling? Endospores. So what if they had endospores? They thought they killed everything, but they didn't. So this is why when you look at and see scientific papers nowadays, they will literally sometimes, so that somebody else doesn't repeat what they did and say, oh, no, that doesn't happen, they'll tell you exactly what brand, what lot number of Petri dishes they used. All the food that they used to feed their organisms, what strain of organisms, the DNA sequence. I mean, any detail they could possibly put in there, they put in there. Right? So that, everything is known. Back in Louis' time, he didn't say exactly what food source he used. We believed he probably used a yeast extract. Others probably maybe used a hay extract. And in that case, it was laying out in the fields and could have picked up endospores from the environment. So it wasn't until these two guys who came along these same answers around the same time in two different places. So they're both given credit. So John Tyndall and Ferdinand Cohen. And they helped demonstrate that it really is the dust in the air that carries the microorganisms. And they showed that if dust was absent, if it was not present, you could leave them completely opened, no swan neck to that flask, and no microbial growth would happen. These are known today as clean rooms, right? Some places, especially where they build electronics and certain medical devices, right, where they have filters that are sucking out and filtering the air, high efficiency filters, HEPA filters, to make sure that there isn't any dust in the air. And then you don't have to worry about any microbe contamination or growth. They also provided evidence for the existence of the extremely heat-resistant forms of bacteria that we now know as what? Endospores. And therefore help people understand why maybe they weren't getting the same result as Louis with the swan neck flower, right? Because they weren't actually killing them by boiling them. They were resistant to that type of heat. So, okay, this is where the microorganisms are coming from. And then, of course, we always come back to the bad news, right? The fact that they can cause disease, right? Make us sick, maybe even potentially kill us. It was not immediately obvious that that's what was wrong for some people that were sick, right? Or in general with illness. That it was actually microorganisms. So a set of techniques had to be developed in order to study microorganisms, okay? So we know they're there, right? We know they can grow. We've got to be able to study them now. And that wasn't necessarily an easy task. I mean, they were using things like slices of potato to grow them on, but I don't know about you, but does everybody in this class like potato? Right? I hate mashed potatoes. I think my mom fed me way too many of them as a baby. She's like, when you were a baby, you loved them. I was like, now I just want to puke. Not all microorganisms like potato, right? So if they don't like potato, are they going to grow? No. So we had to find a way to create a solid surface, to be able to separate them when we grow them, instead of just having them in liquid. But we needed something they were going to eat, too. We needed to give them stuff they needed to eat, but then stuff they're not going to eat, too, so that it'll stay solid. Anybody know what we add to food nowadays to create a solid surface in our Petri dishes so that we can move the organisms around and separate them? Starts with an A. Agar. It is a polysaccharide of sugar derived from marine algae that at this point in time, we don't know a single microorganism that will eat it. So that's good. And it will solidify any of the food that we have and create a nice solid surface to grow them on. Normally I would bring stuff over, but since I'm trucking across campus, I usually bring Petri dishes and stuff in so you guys that aren't taking lab can see this stuff for today. But you're more than welcome, you know, if you're over in Building 1, always come say hi to me in the office, right? Come by the lab. I'll show you stuff if you want to see it if you're not in lab. Let you touch it, let you feel it. The good news is they don't eat it, and unlike another solidifying agent that you use at home to make like your shrimp molds What do you use to solidify a shrimp mold? Gelatin. What is gelatin? It's in Jell-O. What type of molecule do you think it is? Now I said agar's a sugar. What type of molecule? So what are our macromolecules that we're made out of? We have sugars, proteins, fats. Don't forget the most important one that we abbreviate. Go ahead. DNA. Nucleic acids. DNA and RNA. Right? So those four, what do you think? It's not fat. It's not DNA. It's protein. By the way, most of life is protein. Most of functional life, right? The workhorses are proteins. So gelatin is protein. It's protein found in animals. So it is an animal source. So if you're vegan, you don't eat it, right? It's collagen. Yeah, no. Well, I don't know how much collagen's in bone marrow. I mean, they used to say it was ground up horse hooves. I don't know how much validity is to that. But I mean, yeah, it's pretty much any animal source that has a lot of collagen is what gelatin is. But it's protein. So organisms eat it just like we do. So we can't use it to solidify our food because they're going to eat it, most of them, just like we eat it. The other problem is that at room temperature, what happens to it? Is it really solid? No. What do we have to do to make it solid? We put it in the fridge, right? And you leave it out at room temperature, it starts melting. Well, we want to incubate stuff at not even room temperature. We want to incubate it at body temperature. What's going to happen to gelatin at body temperature? It's definitely going to melt. So we needed something that was going to stay solid and other things aren't going to eat it. And so some brilliant, I think actually it was a woman, found out that this particular sugar from marine algae did the trick. So, that took a lot of time and effort and almost even luck, right? To be able to design a way to be able to grow the organism. And then Petri. Why are they called Petri dishes? Because a guy named Petri came up with the design of this dish, right? And if you look at it, you have the bottom part where we stick the food, right? And we add agar to it so it solidifies and we have a solid surface. And then the lid comes up over it, but it overlaps the bottom, right? Brings us back to Louis, because do we want air to get in? We do. Some organisms need it. But do we want it to go directly in? No, we want it to go indirectly in, right? So it's got to go up and over and in. Make sense? So all these things had to come together so that we could start growing them and using them like we do in lab, right? Like you guys will start doing in lab next week. Then it also led to a whole other area of science, my favorite area, is that why is it that not everybody who's exposed to these things gets sick? Immunity, right? We have an immune system that helps protect and fight the microbes that we're constantly exposed to. And this created a little bit of a problem for this next guy, which how many guys went to chapter 18, section 4, and read up on this? Come on, y'all. I told you how to get Adobe a Baccarat Read It read for you, right? So, even if you have trouble reading like me, it will read to you. You need to read. No, remember this one is in chapter 1, but the details about Koch and his experiments is in chapter 18, part 4. I had posted that for you guys, told you last time. And your book tells you when you read in chapter 1 about Koch's postulates, refer to 18.4 for more details. So he established the relationship between Bacillus anthracis and anthrax. He was able to prove that this bacteria, this specific bacteria, caused the disease we know as anthrax. Why is that so important? They didn't know this, right? They didn't know bacteria made you sick. We had trouble convincing them where bacteria came from. They just poof from apple juice. So he set up a set of criteria. But before he could even do this, these techniques, these tools, the Petri dish, agar had to be developed, microscopes. So his set of criteria is known as Koch's postulates. They're still used today to establish the link between a particular microorganism and a particular disease. But even he, as he was going through this, realized that some of the criteria would have limitations. That not all the criteria could be satisfied for all diseases. Say that again? No. You don't need to know that he got it from his teacher. His teacher's not on the list of names you need to know. Koch is. So you need to be able to recognize, right, the description of how Koch's postulates are used to establish the causative link between a suspected microorganism and a disease. And look, right, I know you guys don't have this PowerPoint. I don't think I updated it, but that's the only change I made. This is an 18.4. So this I like because I'm a picture person. I should probably write out the steps on a slide larger for you, but you have them in your book. And I think if you write them out, you might actually remember them. So me doing things for you wouldn't necessarily help you. So the first criteria, which is kind of hard to read on here, but if you just look at the picture, is that you have a poor little mouse that clearly doesn't look like he's doing so well, right? And they're doing what? No, they're actually taking from him, right? And growing it or taking a sample and looking at it under the microscope, observing that they have microorganisms there. So the first is the microorganism must be present in every case of the disease and not in healthy people. Is that true for all disease? Some people get exposed even. And do they get sick? No, because their immune system saves them. So even this one criteria, the first one, can sometimes be a problem for scientists. Not everybody gets sick from it. But each one of the ones that are sick need to have that microbe. And it needs to be the same microbe for us to say that this microbe causes this disease. Right? So all the ones that had anthrax need to have Bacillus anthrax in them. And then they're going to grow it and isolate and make sure they're dealing with just one bacterial species. So that's when we need those techniques. We need those tools, the plates, the agar, the food. And then the technique of actually separating them out. The most commonly used is called the streak plate method. Those you guys in lab will do next week. So that's what you see here is where they've spread them out to try to separate out the bacteria from one another. Generate what we call pure cultures, where you're dealing with a single species of bacteria. So the organism must be grown in pure culture from diseased hosts. Pure means what? One what? One species of bacteria. Not a mixture, right? Not Bacillus anthrax and Bacillus subtilis, right? Just one. Pure means one species. Can all microorganisms be grown in artificial culture outside of an organism? Nope. Can anyone name one that can't? HIV is a virus, so growing is a whole nother story with that one. You know, any organism, an example of one that cannot be grown in artificial culture, has to have a living organism in order to grow. Now, well, actually, we might be right with tuberculosis, but they can grow it in tissue culture. But again, that's not artificial culture per se. You got to grow cells to be able to then grow the bacterium. And so she's on the right track. And I think they talk about Mycobacteria leprae actually in your book. And so I actually worked and did my research with Mycobacteria leprae when I first got out of undergrad at Hansen's Disease Center in Baton Rouge, Louisiana at the vet school. They grow it in armadillos, 9-banded armadillos. And armadillos are also natural carriers. There's some debate on whether it really was in them first or if some of them escaped from the lab years ago. That's what Mycobacteria leprae causes, is leprosy. The PC name for leprosy now, politically correct, is Hansen's disease. Hansen's disease. He was one of the major contributors of, I think, either linking that or the treatment they finally came up with. I should know that, too. I should know what Hansen had did. Well, you can go to the museum at Carville, right outside of Baton Rouge, in St. Gabriel. They have a really awesome museum there. And they still have a few patients that were there at the it was the last colony. I don't know. I can't find the word this morning. It's where basically they sent people with leprosy, right? They weren't allowed to be part of the regular population. Whew, God. We're out of time. Okay. So we will re-look at that next time. And then notice, right? Somebody asked me, do we need to know his teacher's name? Right? Look at one of the next objectives that we'll finish up next time. And get caught up on your reading. Please watch the animation of Graham Stain and all that I posted before next time. These are the names you need to know what they did.